Hi, and welcome to today's program, From Ancient Teosinte to Modern Corn of the Domestication of Plants and People, featuring archaeobotanist Heather Tucker. This is part of our ongoing Hot Topic series. My name is Brianna Pobiner, and I'm a paleoanthropologist and educator at the Smithsonian's National Museum of Natural History. I'm a brown and gray haired woman in a gray checkered shirt wearing headphones in front of a Zoom screen with an African savanna photo with grass and an acacia tree behind me. Whether this is your first time joining us or you've attended our Hot Topics before, we're so glad to have you here. And if you signed up for this event, which was originally scheduled for February and are joining us now, we appreciate you sticking with us. Before we get started, I have a few housekeeping notes. This discussion offers closed captioning. You can turn the captions on or off via the CC button, which should be located at the bottom of the Zoom interface. We're in a webinar format, which means we can't see or hear you. So as you have questions, please go ahead and submit them into the Q&A box. That's at the top or bottom of your screen. It looks like two speech bubbles. And then we can sort through as many as possible because the Q&A time really flies by. The Q&A box is also where we'll share any relevant links during the program, so keep an eye out there. We'll start with a presentation by our speaker, Dr. Heather Tucker, and then I'll join her here to take your questions. During the presentation, I'll also write answers to some of your questions, at least any that I can answer, as well another member of our behind the scenes team, Dr. Ryan McCrae. So now I'd like to go ahead and introduce our speaker. Dr. Heather Tucker is an anthropological archaeologist whose research unites humanities and sciences in the pursuit of knowledge about the human past. With expertise in radiocarbon dating, stable isotope analysis, and the identification of archaeological plant and animal remains, her work advances integration of the archaeological sciences. Drawing on theoretical frameworks of evolutionary ecology, resilience, and traditional ecological knowledge, she seeks to understand the complex evolutionary history of people and plants in the Americas. Her current research focuses on the domestication of plants, transmission of cultural knowledge, and environmental feedbacks throughout the critical region of Central America, supported by ongoing fieldwork in southern Mexico, Belize, Honduras, and Nicaragua. She earned her PhD in anthropology from the University of California, Santa Barbara in 2014, before joining the anthropology department at Texas A&M University in 2016 as the curator for the anthropology research collections, and she is currently a tenure-track assistant professor. So I'm delighted to welcome Heather to the screen to present her research.
And I am equally delighted to join y'all all here today. I am, see if I can follow Brianna's guide. I have dark hair up on top of my head in front of my desk in an office setting and a light colored shirt on. Okay, I'm going to go ahead and share my presentation.
Excellent. All right, take it away.
Fantastic. Well, I'm so glad that people turned out today to hear a little bit about what's going on in the world of domestication research. When I first got started with this, I thought, there's nothing I want to study less than corn, because haven't we studied all of it? It is a major food staple, animal food, biofuel and industrial sweetener, alcohol base. We even use it for bioplastics, right? I grew up here in Texas, driving through massive cornfields and getting out to measure myself in comparison to how tall those cornfields were. It's a part of our identity here. But we're not the only ones, right? The staple grows on every continent except for Antarctica. It provides foods for millions of people. Ancient farmers began cultivating maize's wild ancestor Teosinte about 9,000 years ago. But researchers still wonder why the plant ever became popular, because Teosinte, the ancestral plant, lacks some of corn, or maize as I'm going to call it, maize's most desirable qualities, like an easily harvestable large cob of kernels, right? So we know that the development of agricultural societies between 12,000 and 9,000 years ago, this was one of the most transformative events in human history and in ecological history, right? And it was made possible by plant and animal domestication. Some of you may have been lucky enough to hear the presentation by my colleague, Dr. Angela Perry, in the spring on the domestication of dogs. Together, the domestication of plants and animals really transforms not only human society, but the earth, right? And so even though I thought maybe all the questions about maize had been answered, digging into it deeper, I see that even though we've tried to understand for centuries, literally, the evolution of crops during domestication, making inferences based on our imperfect genetic and archaeological data, population genetic analysis of changes associated with domestication are still limited by the sparse availability of ancient DNA. And the archaeobotanical record itself is often chronologically coarse and geographically uneven. That said, I want to present this conceptualization that crop domestication is an ongoing process, right? It's still happening just as it happened in the past. It's about building and sustaining dynamic coevolutionary relationships between plants and humans. By the time Christopher Columbus arrived, we know that there were at least 200 different subpopulations of maize growing. Its global annual production exceeds 1 billion tons and provides 20% of human calories today. But despite all of this, there's research telling us that we have reason to be concerned, right? How are our modern varieties of maize going to adapt to the environmental constraints brought by climate change? In archaeology, we like to think that the research that we do can contribute to finding solutions. By peering into the history of maize, we ask, could there be genetic variations which could inform new breeding strategies that might improve the future of the crop? And in order to get at those questions, we refer to what you see there as the domestication triangle. Right? This gives us a framework for thinking about that ongoing process of coevolutionary relationships between plants and humans. Right? Starts off with the genetic and phenotypic particularities of a crop plant. Crop diversity itself encompasses diversity within lines and varieties, within the crop as a whole, across the entire crop gene pool, and that's going to include also the wild relatives. In the second grouping in this triangle, human cultural practices, agricultural practices are a significant influence as well, right? Human diversity includes all of the tools, the interventions used in domestication, agricultural products and forms of labor, including both the culture and the cuisine that drive domestication. In that third part of the triangle, we're looking at the role of ecological and geographical features. So ecological diversity spans from the level of a single field to the agroecosystems, which might have multiple fields and their surrounding areas, or it can expand to regions and landscapes. So thinking about the complexity of ongoing crop domestication as a part of these processes, interlocking, right? All three are elements, crops, humans, and ecosystems, that are important to help us explain how agriculture resulted in the major transitions in human history, how it facilitated the rise of a dominant contemporary society and food system. And beyond that, looking towards understanding how crop domestication intensified production in ways that it could support large populations. At the same time, it also contributed to the depletion of genetic, agricultural, dietary diversity, and there have been subsequent negative ecological and social impacts. Right. On this slide, you'll see a picture of the book Endangered Maize, an excellent read written by science historian Helen Curry. One of the things she asks is, how are we going to handle threats to the diversity of maize, given its importance in our economy, in our cuisine, in every aspect of our lives these days? So thinking of domestication in this way, as the relationship between crops, ecosystems, and humans, it makes it clear that the challenges that we face in modern food systems, creating crops that are resilient and sustainable, nutritional and equitable, right? It lies at the intersection of these elements. And we can really only overcome it by engaging factors from across all of it. So this is where we turn to our broader perspective coming from information we get in the archaeological record. The oldest maize cobs in the New World are from Gila and Iquitos. This is a cave in Oaxaca in southern Mexico. The cob you see at the bottom is only about 2.5 centimeters long. The one at the top is a fragment that's even a little bit smaller. These have been directly dated, the older one to 6,250 years ago. The history of modern day maize, we know, begins with the dawn of human agriculture. Ancient farmers in what's now Mexico took the first steps in domesticating maize when they simply chose which kernels to harvest, which kernels to plant. They noticed probably that all plants are not the same. Some plants may have grown larger than others, or maybe some kernels tasted better or were easier to grind. The farmers themselves saved the kernels from the plants that had desirable characteristics and planted them for the next season's harvest. This process is known as selective breeding or artificial selection. Maize cobs steadily became larger over time with more rows of kernels, eventually taking the form of something that we would all recognize today as modern maize. Here you see two drawings. One is of the ancestral plant Teosinte. Specifically, it's Zea mays parviglumis, which is one of the two Teosintes that contributed significantly to the modern genetics of Zea mays maize, which is corn as we know it. So during domestication, crops evolved a suite of phenotypic traits. We often call these the domestication syndrome. And these characteristics are what we use to distinguish crops, cultivated plants from their wild ancestors or wild relatives. Some of the modifications we see include giantism in the harvested plant part. That's going to include fruit enlargement, seed and seed head enlargement, leaf enlargement, increased fruit size relative to leaf size. And there's an obvious benefit there. The part that humans like is the one that they're selecting for to be larger. We also see a tendency towards reduced branching. So if you look closely at the difference between the Teosinte and the maize stalk, right? The shift tends towards a more clustered instead of a branched seed head. There are also changes in root form, all of which contributes to having simultaneous ripening. Right? All of the corn cobs on a modern plant are going to ripen at relatively the same time. We also see a loss of shattering. That means that those corn kernels really stick into the cob. There's more seed retention. In the teosinte, it's a brittle connection, so it's more likely that they'll be able to shatter and spread out. We also see the loss of protective structures like a reduced seed coat thickness. So between domesticated maize and its wild ancestor teosinte, we specifically see that modern teosinte has long primary lateral branches tipped by tassels and secondary branches where female inflorescences are located with just a few small seeds covered by hard fruit cases that mature sequentially over a period of a few months. In contrast, maize has a single stem that terminates in a solitary tassel. They're just a few dramatically shortened lateral branches and no lateral branching. The cobs are located at the end of the branches in the main stem in the positions occupied by tassels in the teosinte. Maize seeds are not covered by fruit cases and its cobs mature about the same time. Okay, so these differences are among the most dramatic that are documented for any major crop or ancestor pair. And it's led to a century-long debate about maize ancestry. Some of the modern domestication research has focused largely on the identification of morphological and genetic differences between extant populations of crops and their wild relatives. Less attention has been paid to the potential effects of the environment, despite substantial known changes in climate from the time of domestication to the modern day. So in one of my all-time favorite experimental studies led by Dr. Dolores Papirno at the Smithsonian Tropical Research Institute, they took the wild ancestor of maize, teosinte, to environments similar at the time of domestication. When the primary goal of the research was to evaluate the role of phenotypic plasticity in the evolution of maize during its domestication. So when I say phenotypic plasticity, this is a subject of growing importance in evolutionary biology. And recent research has shown that gene expression is key to understanding both the plastic and the adaptive responses of plants to changing environmental conditions. So to better understand the potential role of the environment in maize domestication, they took the two different plants and grew them in two very different environmental conditions, one reflecting more modern climates, one reflecting what we think the ancient climate would have been like under the lower temperature and CO2 conditions that match with the terminal Pleistocene and the early Holocene. So both this effort demonstrated that there's a really dramatic phenotypic change in teosinte when it's grown under ancient conditions. That includes a lot more plasticity in the gene expression and more substantial changes in the expressional networks in teosinte across different environments. Additional studies since then have identified gene set of over 2,000 loci showing a change in expression across the environmental conditions in teosinte and in variants in maize. So those 2,000 loci allow the ancestral teosinte to be very flexible in the environment, but they become fixed in our modern domesticated maize. Right? And so these results suggest to us that early agriculturalists may have selected for genetic mechanisms that cemented this domestication phenotype that was initially simply a plastic response of teosinte to the environment. If we look at the phylogenetic tree for the Zea genus, right, These results add to a growing body of literature that identifies the important, if not dominant role, that admixture also played in the history of domestic plants, including maize. Earlier studies were limited to the non-recombining components of plant genomes, but these approaches didn't give us the full interweaving branches that result from admixture. And so they weren't really sufficient until recently to help us address or quantify this idea of admixture gene flow between populations through time. In this chart, you see the divergence times estimated for multi-species coalescent model. The arrows indicate the taxa involved. So a one-way arrow indicates a unidirectional introgression, whereas a two-way arrow indicates a bidirectional introgression. And the arrow color shows the value of Patterson's D statistic. Morphological characteristics of Teosintes, particularly nicaraguensis and luxurians, are separated out from all the others. And they are here treated as an outgrouping. The Mexican annual Teosintes of Parviglumis and Mexicana are distinguished from each other based on the presence of macro hairs and pigment along their stems. Both of these traits are linked to a highland adaptation. So in looking at the modern maize and the extant Teosinte, we've actually been able to learn a lot about the relationship between these taxa. We also consider the natural geographic distribution of Teosinte, which extends from the western Sierra Madre in the state of Chihuahua to the Pacific coast of Nicaragua and Costa Rica, including practically the entire western part of Mesoamerica. Mexican annuals, like Parviglumis and Mexicana, show a wide distribution in Mexico. Other types of Teosinte have a much more restricted and distinct range. This has drawn our attention to the two Teosintes, Mexicana and Parviglumis, not only for their wide distribution, but because they exhibit really different altitudinal distributions. Parviglumis likes to live in subhumid tropical environments at lower elevations, but Mexicana likes to live in subtropical to temperate conditions in the highlands from 1,500 to almost 3,000 meters above sea level. So our growing appreciation that both Parviglumis and Mexicana had significant admixture with maize, we have come up with a variety of different types of demographic domestication models. There could have been a single domestication with ongoing gene flow, multiple domestications and improvement events with gene flow, and then another version of that in 6. Right? Multiple domestications from the same ancestor. This is a process that we know was complex. We know there was recurrent gene flow between different cultivars and their wild ancestors, but trying to understand the exact nature of that and how we ended up with the particular varieties that we ended up with requires us to directly assess the genomes of ancient populations through time. And that's not always easy to do. But I do want to go ahead and shift and talk a little bit about the data that we do have from archaeological sites. So here, I'm not just talking about the domestication of maize, right? I'm wanting to dig in a little bit more deeply to the diversification of phenotypes, right? What are the trends in the nature of genetic variation? So in this context, domestication genes are those that are going to lead to phenotypic differentiation between a crop and its wild ancestor. And diversification genes are those that are going to result in phenotypic variation within a crop species, maybe even between different varieties. So as the evolution of domesticated plant species proceeded, it led not only to genetic differentiation between the species and their wild ancestors, but also to an increased phenotypic variation. Within the species, it showed up as new traits that could be selected for. That's the diversification part. Now, because we do see a lot of diversification, the identity of maize's wild ancestor was a mystery for a very long time. This is one of the hot topics of argument in the past. Looking at some of the evidence more directly, I want to tell you where we see some of the first evidence for early maize domestication. So the earliest cob in the assemblage here from El Gigante, right, that occurs during a pre-ceramic phase. It was directly dated to 4,340 cal BP, so approximately 6,000 years ago. No, 4,000 years ago. It has 12 rows with paired spikelets at the base of each long, deep cupule. It has gloom induration. So remember those thicker seed coats associated with Teosinte, and thickening that's consistent with the relatively recent genetic contributions of Teosinte at the time of domestication. So what we see showing up here at El Gigante is fully domesticated maize, but it still bears some of the characteristics of its ancestral Teosinte. Right? Other dates for the initial recovery of maize we have from the Ocampo caves in Tamaulipas, the Tehuacan Valley, Gila Niquites, and then all the way down south in Peru. So one of the other sites we'll take a look at is Perdones. Starting off in the north, in the Tehuacan Valley, right, this is where we have the oldest known cob. This is believed to be the center of domestication. Both Parviglumis and Mexicana have been recovered from the region, and there seems to have been genetic contributions of both. More recent excavations in the Tehuacan Valley, specifically here at San Marcos Cave, right, has produced a large collection of maize macro specimens. They've been collected from archaeological sites, but only a few have been directly dated. So one of the newer approaches is to directly date each and every maize cob so that we can associate morphological genetic phenotypic changes with a timeline across space. So direct AMS dates for samples here from San Marcos confirm that the reported chronologies are pretty robust. So that early specimen at 5,000 years ago is supported by the abundance of dates of other specimens that are similar in age. If we look at a comparison of all of our chronological dates for macrobotanical remains or maize cobs or kernels themselves, it doesn't really reflect that the dispersal had to go from central Mexico to the north or southern regions. There's the possibility that we might still find missing links in further investigations within Mexico and Central America. We also see in these excavations from the San Marcos Cave specific evidence of root traits that were related to maize domestication. Using a specialized form of tomography, The authors here were able to characterize the root architecture of a 5,300-year-old maize specimen recovered from San Marcos and get a good look at the incredibly well-preserved cellular organization. The outer cortical cells in these pictures contain thick lignified walls. That's typical of extant maize, adapted to hard soils. By contrast, there's an absence of seminal roots. This is only found in the maize ancestor, Teosinte. The genes for seminal root development had mutations that could relate to their absence, but the collective results indicate that traits evolving in the root structure of maize were related to drought adaptation, and they weren't fully present in the earliest forms of maize at Tehuacan. So this is a line of evidence suggesting that we continue to see the process of domestication unfolding even once maize becomes what we call Zea maize and not Teosinte. If we look at the evolutionary relationships between the ancient Tehuacan maize and its wild or cultivated relatives, here you can see the Teosinte group is highlighted in green. The maize landrace group is in red, and ancient maize samples from San Marcos are in blue. So right there in the middle. This reevaluation of the DNA sequenced from these cobs, all are about a similar age of 5,000 years old. They reveal a whole new level of genetic diversity compared to modern maize, right? It indicates that the effects of domestication were not yet complete. Right? Their genetic constitution was similar and influenced by inbreeding, and corresponding plants seem to have come from a reduced population, isolated, perhaps self-pollinated individuals. So through the genetics, we're able to learn not only about the ancestral relationships, but the ways in which these populations were also managed by humans. We do know that Native Americans domesticated maize from lowland Teosinte parviglumis in the warm Mexican Southwest, and then it was later brought to the highlands of Mexico and South America, where it was exposed to lower temperatures that imposed a strong selection on flowering time. One of the most interesting developments is evidence of adaptive Teosinte mexicana introgression. So mutations related to highland adaptations appear to be the result of mexicana introgression into maize, which improved its fitness by accelerating flowering. If we go all the way down to Peru, also 5,000-year-old corn, right? We can look at the relationship between relatively contemporary specimens from central Mexico and Peru. So the archaeological cobs here that you see are from Perdones and Huaca Prieta in Peru. They represent some of the oldest maize known to date, but they also present phenotypic traits that correspond with domesticated maize. It's apparent that this is fully domesticated maize, showing up in Peru 6,700 BP. Right? This suggests that there's a really rapid dispersal followed by improvement as maize was carried from the central Mexico region into South America. Right? Even more interesting is that those oldest individuals do not have the introgression of mexicana. Right? It has very few alleles that have been previously found to be associated with highland varieties. and it looks like they may have been associated with a lowland migration route. So overall, the data that we're getting out of Perdonis is that the maize originated in Mesoamerica, like we've thought for a while. It arrived in Peru without that introgression from Mexicana through a rapid lowland migration route. And then it went rapid under improvements, both in Mesoamerica and South America. Now I'm going to take you over to Western Honduras, where we have a newer rock shelter that has only more recently been studied. This site has tens of thousands of very well-preserved, desiccated plant remains, including tens of thousands of maize cobs. Okay. We have a clear chronology, as you can see here, that not only allows us to look at the abundance of maize in the people's diet, but also how it changes in relationship to the use of other plants. It's not until the late Archaic that maize shows up at all in the record at El Gigante. But once it does, it takes off in the early formative, becoming one of the primary constituents at this site. So archaeogenomic studies have suggested that there's a process of protracted maize domestication with multiple waves of human-mediated dispersal into the Americas. It first travels south into South America. According to some studies, this may have been a partial domesticate, but it's fully domesticated by the time it gets down to Peru. Based on the genetics of cobs that we have excavated from El Gigante, it's clear that the domestication syndrome becomes independently fixed and improved while away from the natural range of Mexicana and Parviglumis. So in this study of the ancient El Gigante maize and its cultivated relatives, we hypothesize that this backflow of South American genetic material may have contributed to the development of a more productive staple, which was related to the growth and aggregation of human populations and the formation of more complex social and political structures, right? More productive maize through this process of diversification actually enables the development of much more complex social forms as people are able to sustain larger populations. Knowing this coming from the genetic data, we can also look at the morphological variability in ancient maize at El Gigante. Right? The preserved cobs from the rock shelter have been directly dated, and there's an extreme variation in size, row number, kernel count, rachis width, the type of cupules there are, whether they are hairy or not. And these are traits that we know correspond with the degree of Parviglumis or Mexicana ancestry and the degree to which Teosinte was a recent component of the genetics. So looking at over 10,000 carbonized and uncarbonized maize specimens is one of the things that we are doing right now, and directly dating all of the ones that are distinct in their morphology and making sure that we are trying to understand what those differences are telling us, even if we do not have full preservation that is good enough to allow for DNA extraction. We do know that there are temporal differences in the cupule width and the cupule wing width. This corresponds with the size of kernel that would have been in the cob. Right? The shift suggests that the attributes on average became much larger, suggesting a selection for increasing seed size. This has been robustly repeated as one of the characteristics that we see. We also look at differences not only in those reproductive characteristics, but the vegetative characteristics. So looking back at this record, studying the diversity, the morphological variability of ancient maize at El Gigante, we hope is going to give us greater insight into that domestication triangle. Because at an individual point in time, given the density of this record, we're able to say, what were all the varieties? How did they relate to each other? Is the differences that we see related to environmental variation, like increased fertilization or polycropping, are the differences that we see related to human intervention? Is it related to climatic change, rainfall, drought? So looking across a known synchronous set of cobs from this assemblage, we actually get to start talking about where these varieties came from, how they were introduced, which ones persisted and why. And we know that that local engagement in the domestication process is really significant for maize. Right, this is an example of the 59 native Mexican land races that are extant today. Mexican agriculture today is very heterogeneous because farmers keep growing these local land races. There's sensory, aesthetic, ritualistic, and cultural values associated with it. Now, today, I don't think that our goal has to be to keep land races in a state of stasis. It's probably not possible and perhaps even not desirable. But it is possible to promote efforts to preserve wild and domesticated gene pools, both ancient and modern, to allow for the continued evolution of this important crop. Efforts that include improving local land races, encouraging seed saver networks, developing markets for land races that recognize their added value and the evolutionary services of small-scale farmers can all contribute to how we deal with this ongoing adaptation of crops to climate change. So looking back into the past, we can use that to help prioritize different types of crop diversity. The species that make up our current food systems are really homogeneous. It's estimated that tens of thousands of plant species are edible to humans, but only about 2,500 species from only 170 different taxonomic families have actually undergone domestication. Right? Only 300 of those are even considered fully domesticated. So we can look to the past to leverage this diversity of plant forms that did exist and help us to better understand the life history and functional traits that support ecosystem services important to us today. We can also introduce new diversity into existing major crops. Part of the classic domestication syndrome, or the suite of phenotypic traits associated with domestication, includes larger seed size, a loss of seed shattering, or reduction of lateral branching. Collectively, this makes harvesting easier, but it also results in undesirable changes like the loss of genetic diversity, right? The accumulation of mutations that may not be beneficial, and changes in the metabolic profiles of the food itself. So the challenge ahead, if we want to ensure that our crops are resilient, sustainable, nutritional, and equitable, is to try to find new ways of increasing that diversity or reducing the mutation load. These can help increase our overall crop resilience while maintaining important traits that have been acquired throughout the process of domestication. So I'm going to go ahead and pause there because I have many more slides and I could talk a lot more about maize, but I think we're probably at a good place to start addressing questions. And I do want to share my last slide for my department, which has been incredibly supportive of my multi-pronged methodological approach to trying to understand the process of domestication.
Fantastic. Thank you, Heather. And we have a bunch of questions already, so this is really good timing, I think, for a stopping point. I'm going to start with the first question that came in, which asks a creative question that I don't know if there's, if anyone has looked for this, but Sophie asked, do any of the ancient cobs found have clear human teeth marks?
Oh my gosh, that is a really fun question. So we have cobs that have clearly been popped, popcorn. We have cobs with the kernels still in them. I have never seen teeth marks, and part of that is probably due to the cob architecture itself, not being able to preserve that really well. There's always the chance, though. That's the beauty of archaeology, is it could happen.
Excellent. And I'm going to actually jump to a follow-up question, or a different question from the same person, from Sophie. Are there preserved cobs with the kernels in place? And if so, is the color of the kernels preserved?
So yes, I have preserved kernels. And I'm talking specifically about El Gigante because that's what I have here in my lab right now, are all those maize cobs and kernels. So I find individual kernels. I also find kernels embedded in the cob. And we measure those just as we do the cob to get a three-dimensional morphometric understanding of changes in the shape and structure of the kernels. Sometimes color preserves, but that's not something that preserves exceptionally well. And I would say the variation that I see in color is more related to the preservation conditions of the cave than it is the actual original color.
That actually follows beautifully onto this next question from John, who asks, are there special environmental conditions that preserve these 5,000-year-old cobs?
Yes, you might have noticed all those places I was talking about, rock shelters and caves. So any kind of organic material is very likely to decay out in the open environment. But if you give it a nice, dry, desiccating environment, an anaerobic environment that inhibits that decay, we can actually get preservation for, in this case at El Gigante, we're looking at preservation of plant remains for the past 12,000 years, right? Not corn, not maize, that doesn't show up until later, but we do have very ancient plant remains because it was a dry, desiccating environment protected within the rock shelter.
Great. So here's an interesting question that dives more into the genetics from Lynn. Looking at maize genomes over time, have you seen changes in structural variations, such as increase or decrease of mobile elements and chromosomes that duplicate and or disappear?
Yes. And this is not my research, but there's an extensive amount of work that has been done with modern and ancient maize genetics. One of the most consistent things that we see, though, is in fact a reduction in the size of the genome related to that domestication process. So as more characteristics become fixed, there's less plasticity, there are fewer novel traits being introduced. So we do see a pretty significant reduction in that diversity. There are absolutely specific mutations, like related to sugar development, that we see associated with changes in specific varieties of maize. So yes, that's there. There's a significant literature available on it. And unfortunately, that is not entirely in my wheelhouse.
And this next question, which is you've brought up some points that may have answered it, may not be either. But Linda asks, can you address the varying traits and the likelihood that they were intentionally selected for or unintentionally emerged from the context? I don't know if there's a way to tell that.
This is something I think about a lot.
Mhm.
And it comes to this point where we have integration of a lot of different archaeological sciences. So one of the things that we do is, okay, I've measured all the cobs, all of them. Every cupule, the length, the width, the shape. I've done the same for the kernels. We have that data. We also have environmental data that we can bring into bear. We also have, one of the things we're working on right now is isotopic data directly from the maize itself. That can tell us if there are nitrogen enhancing fertilizers being added to the soil. It doesn't necessarily tell us whether those are being added naturally or by humans, but we can see if there is increased fertilization of the crops. We can see, based on the isotopic record, if there's increased bioavailability of water. Humans could be an agent involved in both of those. So then what we look at are, what are the differences in the specific traits that are changing at a given point in time? Because vegetative traits are more likely to respond to environmental change, right? These are the ones that are going to demonstrate a lot of phenotypic plasticity, like the branching of the plant. That's in contrast to the morphological variation of reproductive characteristics. Those are the ones that humans are selecting for. You know, the kernel size, the kernel thickness, the kernel characteristics, right? That's direct human intervention and a better sign for selection. So we want to look at all these lines of evidence together. Even if I can't get DNA, right? I can look at the isotopes. I can understand what are the changes in the environment that the plant is being exposed to. And I can look at what are the contemporary changes in the morphology. Are they related to the environment, or are they more likely to be related to human interventions?
Here, I'm going to switch tacks a little bit to a sort of practical methods question. So Rosemary asks, how do you preserve in your lab the excavated samples of corn? Freezing them?
Freezing is a good option. I prefer keeping these. All right, so I'm going to back up. It depends on the specific preservation context that your assemblage is coming from. So if I was going to work with Utsi, right, the frozen person in the Alps, preservation would require me keeping him frozen. So freezing, yes, that's one way. I keep pollen samples frozen in the refrigerator because I don't want microbial growth to actually damage the pollen. For these samples, right, they were preserved in a dry, desiccating environment. That means the thing most harmful to them is going to be humidity. So I could freeze them, but successive freezing and thawing is not good for the plant, and that wasn't part of its original preservation context. Being in a dry, desiccating environment is. So I make sure that they are packed in a way that there is minimal humidity to accelerate that decomposition.
Okay, great. Thank you. Here's a broader question from Stephen. You commented that maize was cultivated worldwide. Why was it not as popular, he put in quotes, in Europe, Africa, and Asia?
Well, maize is currently grown worldwide, but in the past, it was only domesticated here in the Americas. It only spread to other parts of the world after the period of colonialism, right? That was a part of the process that took a number of Mesoamerican crops like tomatoes and avocados and chocolate all over the world, right?
And actually, here's a question that follows on that a little bit, although it's quite broad. Urate asks, how has maize changed or diversified as it went from the Americas to the rest of the world, especially Africa?
That is a great question. I've actually seen some papers that do evaluate that, particularly maize's response to being in an environment with different types of noxious weeds. But to tell you the truth, modern botany is again somewhat outside of my wheelhouse, so I haven't kept up with that. I try to make sure I have my finger on the pulse of whatever is happening with ancient maize. And in the last decade, that has been more than enough to keep me busy.
That's fair. We'll have time for just a couple more questions. One from John, back to color. The contemporary varieties have a wide variety of kernel colors. Is this the result of human intervention? If not, what's the evolutionary advantage of various colors?
Yeah, the variation in kernel color, first of all, there's inherently a genetic component. That means that the ancestral plant had to have the capacity to produce all those different colors, right? We know that some teosintes have very white kernels and some have much more orangey yellow kernels. And there's a variation in the range of colors we see in teosinte. So that basis existed. Then you take that plant, right, and you start growing it somewhere else. You have different environmental conditions, different soil conditions, new microbial communities, right? New water conditions. All of that is going to influence the way in which those color genes are expressed. So when you start growing a plant in a new place, it may actually start to produce a different color of kernel because it is in a different environment. And that phenotypic plasticity that is allowed for in the ancestral teosinte can allow for variation in color to show up in ways that it didn't before. And the genes were always there, they weren't expressed. And under the right conditions, they are expressed. At that point, people can select for it. And they did.
And I guess, one thing I often think about is that it doesn't mean that there was necessarily an evolutionary advantage just because we see like a variety of color expressed. It might just be, like you said, either preference or just that phenotypic plasticity.
That
all right.
fixed.
Yeah,
right?
exactly. All right, two more questions. One is from Urate again, who says, great talk. What does it mean for a crop to be equitable?
That's an excellent question. So when we're talking about equitability, particularly with modern crops like maize, part of that argument is that land races in Mexico, which are typically locally grown and maintained, there's an incredible amount of variation, right? This goes back to me saying that it's a very heterogeneous crop at that small scale farming level. Well, the long-term genetic lineages can be undermined when modern varieties are brought in and cross-pollinating with those land races, right? So modern commercial varieties of maize may undermine the preservation of some of these more rural land races that are in fact the ones that people actually depend on. They're the ones that they're growing to make their own food, to feed their family.
That's helpful. Thank you. Last question from Walter. Can you recommend a comprehensive and readable book on modern day agriculture?
Oh, on modern day agriculture.
Or even
Again,
ancient agriculture,
I think
maybe.
we're going to have to back up the truck a little bit. My knowledge base tapers out significantly once we get into the current era. In terms of ancient agriculture, or at least maize. I'm looking at my bookshelf right now.
Excellent.
And there are too many really lovely books. I'm going to mention one in particular that I like, written by Annabelle Ford, and it is about the Mayan forest gardens. So the way in which lots of tree crops and other food resources are grown in traditional Mayan communities, and how we see that reflected in the archaeological record as well.
Excellent. You said the author was Annabelle Ford?
Annabelle Ford, and I think, yeah.
Okay, excellent.
Trying to remember the exact name, but I can't remember it.
No worries. Maybe we'll see if we can send that out with,
Sure.
you know,
I can give you a
afterwards.
list of books in general.
Oh, that would be wonderful.
That's my
Thank you.
favorite thing.
Excellent. Okay, so we are going to wrap up. Thank you so much, Heather. We'll conclude today's virtual program. So please join me, everyone, in thanking Heather for sharing her work with us. I'd also like to give special thanks to those who made this program possible. That's our behind the scenes team who helps sort through your questions, including Dr. Ryan McRae, who works with us at the Human Origins Program, to our donors, volunteers, and viewers like you. And finally, to all our partners who help us reach, educate, and empower millions of people around the world today and every day. We thank you. Next month, join us for our next hot topic on October 14th at 11:30 a.m. Less is more, the inclusion of small game in ancient human diets, another diet-related talk with Dr. Grace Veach, who's a postdoctoral fellow with us at the Smithsonian in the Human Origins Program. This program was also rescheduled from earlier this year. We've put a link in the Q&A where you can find more information about our upcoming programs and how to sign up for the museum's weekly e-newsletter. That's the best way to stay informed on upcoming programs and learn more about the museum's research and exhibitions. And all of our hard topics for the fall, so October, November, and December, have all been scheduled. So if you want to find information about that, you can also look on the Natural History Museum's website. After this webinar ends, you'll see a survey pop up asking for some feedback about the program. Please take a moment to respond. We're very curious to know what topics you might be interested in seeing for future programs, and we appreciate your input. So again, thank you, Heather. Thank you, audience, and we'll see you next month.